Hello, my name's Richard Bateman and I'm an orchid enthusiast based at Kew. It's a pleasure to be invited to speak to you today about my favourite obsession, our native orchids. I've chosen to focus on Helleborines of the genus Epipactis, partly because their biology is interesting in so many ways, and partly because they've proved to be taxonomically controversial, especially so in northern England. Following a brief introduction, I've divided the talk into two parts. The first and longer part discusses the evolutionary origin of species in the genus, in terms of a supposed distinction between species that specialise in cross-fertilisation, known as allogams, and species that specialise in self-fertilisation, known as autogams. These contrasting modes of reproduction have profound consequences for the genetics of these species, and it's genetic evidence that I'll be presenting as the central theme of this talk. The second part of the talk focuses in on some recent taxonomic controversies affecting British Epipactis, particularly those relevant to Northumbria. Finally, I'll close with a few higher level conclusions. But first, some basic background. A typical plant cell consists of not just one, but three genomes. Most of you will be familiar with the concept that our genetics is dictated largely by chromosomes held in the nucleus of the cell. And the vast majority of our appearance and arguably our behavior is dictated by those genes. But there are also organelles within the cell that can similarly be quite informative about the relationships among species and how they originated. And in the case of plants, these genomes occur in the chloroplast, which is responsible for photosynthesis, and the mitochondrium, which is responsible for respiration. And the chloroplasts in plants are particularly useful in this regard. The key thing to recall is that the genome in the nucleus is inherited equally from both parents, whereas information from the chloroplast is inherited only from the mother, only from the seed parent. Most of the information I'll be talking about is based on DNA. And of course, DNA is a language of just 4 letters, A, C, G, and T, arguably a 5th letter if you include gaps in the genome. So very simple, basic language allows extremely complex expression of that language in order to generate the appearance of different species. In terms of actually analysing the genome, I would say that the kind of approaches that have been applied to many organisms, not least Epipactis, fall into 3 main groups. The first is protein analysis, which used to be performed on gels in the period 1970 up to the end of the last century. And this kind of analysis is quite challenging to conduct, and the range of variables that you get that will differ between species is pretty low. It's generally less than 30, sometimes much less than 30. And that kind of analysis is now considered to be pretty archaic. That was replaced by direct sequencing the genome to generate the letters A, C, G, and T in the order in which they appear in the genome. That's known as Sanger sequencing and caused a revolution in our understanding of the relationships between different species and how they evolved. And a typical analysis of that kind, based on the Sanger sequencing, looks at individual genes, picks them out from the genome, and sequences them to generate somewhere between 30 and 200 variable sites as a rule. That kind of work started to be applied to orchids at the end of the last century and continues today, although it's the respect with which it's viewed is beginning to decrease as it becomes replaced with next generation sequencing, where basically much, if not the entire genome, is sequenced, generating thousands to tens of thousands of variables. And these kind of techniques have been applied to the orchid family from the last 5 years or so. And the bulk of the information I'll be talking about today is based on that third highest level category of analysis. I need to say a little bit more about the relationship between autogamy and allogamy, although it's surface value, it's relatively straightforward. In the case of allogamy, you're typically transferring pollen grains from one flower to a flower of an adjacent plant. So in doing so, you're mixing the genomes of two different individuals, which of course keeps the gene pool refreshed and dynamic. Whereas if you indulge in autogamy and the pollen grains of a flower land on the stigma of the same flower, causing self-fertilisation, then that mixing of genomes is not taking place. And it allows, amongst other consequences, is that it allows deleterious mutations, injurious mutations to accumulate, rather than being mixed in with other more beneficial versions of that same gene. This, of course, is why our laws forbid us from marrying anyone closer to us than a cousin. So, at least in classical genetic theory, allogamy has advantages over autogamy, and the majority of flowering plants seem to have evolved specifically to encourage allogamy. However, that is not the case with a large percentage of species of Helleborine, of Helleborines. One of the problems we have, particularly in the genus Epipactis, but also in other European orchid genera at the present time, is taxonomic oversplitting. At the present time, as best I can judge, there are about 70 current names referring to Epipactis, subgenus Epipactis in Europe and Asia Minor, and several more are described each year. They're typically described without any kind of genetic or morphological analysis. They're described using techniques, so-called techniques that have existed for the last 3 or 4 centuries. And they're often described by amateurs. If you actually apply these modern cutting-edge DNA-based techniques to these taxa, you end up concluding that there are around about 20 genetically distinct species rather than the 70 species that some traditional taxonomists would have you believe. But let's simplify things and just focus in on the British and Irish taxa. That should be a lot more straightforward because on present evidence, we have only 7 species of Epipactis in the British flora. Epipactis palustris, the marsh helleborine, is pretty distinct, shown here in the bottom right-hand corner. But the remaining taxa are all closely related. And they're a mixture of species that have long been viewed as dominantly allogamous, which include Epipactis helleborine there at the top, and Epipactis purpurata and Epipactis atrorubans at the bottom of this diagram. Whereas the three species that I've shown in the middle of this diagram, Junensis, Leptochila, and Phylanthes, are traditionally viewed as being autogamous, and in many people's opinions, wholly autogamous. In other words, the flowers only self-fertilize, they don't cross-fertilize. And I'll be using genetic techniques during this talk to explore whether that dichotomy between self-fertilizers and cross-fertilizers is actually justified by genetic evidence. So let's take a closer look at the business end of an Epipactis. There's a close-up of its sexual organs. In this particular case, we're looking at Epipactis purpurata, which is supposedly an allogam, a cross-fertilizer. To orient you on this diagram, we're looking at the reproductive organs from the side. The pollinaria are the pollen masses. In orchids, as many of you will know, the pollen is aggregated. Vast numbers of pollen grains are aggregated into tight masses, which are usually removed in their entirety by pollinating insects and transferred to another flower. And in this case, they're protected underneath an anthocat, which, amongst other things, keeps the rain off. The stigmatic surface is underneath this structure, and it's separated from the pollen masses by an adhesive visidium mounted on the rostellum, which you can see poking out towards the bottom right. And that structure plays a key role in keeping the pollen masses away from the stigmatic surface of the same flower. What if we compare a typical cross-fertilized Epipactis like Epipactis helleborine, the widespread common Helleborine, shown here on the left, and a supposed autogamous taxon such as Epipactis leptochila here on the right? And one of the things that you'll notice is, if you look at the diagram on the right here, you'll see that the pollen mass looks far more friable, far less cohesive than does the pollen mass on the left, and pointed out here by a yellow arrow. And you'll also see that the visidium, that glob of sticky glue that is intended to attach the pollen masses to a visiting insect, typically a wasp, is much more reduced in the right-hand diagram, shown by a white arrow. So there do appear to be subtle morphological differences between the autogams here on the right and the allogams over here on the left. And you can see how it might be possible for the flower on the right, for those friable pollen grains to fall onto the stigma of the same flower. What about genetic evidence in these Helleborines? Well, I'm going to begin by just showing one diagram of protein information. In other words, the most primitive of the three techniques we could apply to this problem, that Pete Hollingsworth and I acquired in the latest 1990s and earliest noughties. And you'll notice here that there's sort of microspecies names here shown under traditional names, and then a series of columns here that contain coloured boxes. Now those columns relate to the products of different genes. Each column is a different gene product, and beneath them are different variants of that gene product. The details of this aren't important, but you can see at a glance that the taxa at the top of this diagram, Epipactis helleborine, purpurata, and atrorubans, which we believe to be allogams, cross-fertilizers, have a diversity of different products for each gene. In other words, they have greater genetic diversity than the taxa towards the bottom of this diagram, like Epipactis phylanthes, Epipactis leptochila, and Epipactis junensis, where there is a consistent genetic composition and no evident variability. And initially, this was how genetically we confirmed where the taxa were allogams, these uppermost taxa, or autogams, the lowermost taxa. But that's a pretty crude approach, and you can see how few variables we have to make those kind of assertions. I won't be talking about this style of data anymore. Instead, I'll be focusing the remainder of this talk on this particular analysis, which we ran a couple of years ago using the cutting-edge RADseq technique, a next-generation sequencing technique, which, as I've shown in this red oval, generated 12,543 variable sites, variable characters that we can use to reconstruct the evolutionary history of these supposed species. Now, in order to read one of these evolutionary trees, which we're essentially looking at from the side, you need to realize that the bottom of the tree are the oldest speciation events. That's when species back in time separated and first became distinct from the remainder of Epipactis. And then the species shown on the top right of these diagrams are the ones that originated most recently. In order to recognize these groups of plants as a species, they need to have just one branch at their base. And ideally, that branch needs to be relatively long. So, for example, if you look at Epipactis phylanthes, the pendulous flowered Helleborine, in the bottom left-hand corner of this diagram, you'll see there are several different plants that have been enclosed in a green box. And there's a nice long branch stretches out to the left from that green box. That's telling us that all of those individual plants belong to the same species and have a single evolutionary origin. One of the interesting things in this case is that Epipactis phylanthes, which of course is a British species, is the same thing as the continental species Epipactis persica and Epipactis exilis. At some point, taxonomists have said that persica is a distinct species and exilis is a distinct species, whereas this analysis is telling us that is not the case. All of those continental taxa actually belong within Epipactis phylanthes. So that's how you analyze one of these evolutionary trees. You're looking at the way the tree branches towards the tips. And all of the boxes that I've marked in green are genuine species, the British ones being marked with asterisks. And where there are taxa that are beginning to diverge and look as though they may one day become species, but they haven't yet diverged sufficiently to do so, I have marked those in blue. And you'll see some familiar names here. One of the things I would point out is the considerable evolutionary distance between Epipactis junensis in the top right-hand corner of this diagram and the remainder of the British species, which are on the left-hand side of this diagram and originated much earlier on. Something else that's worth noting in this diagram is that all of the individuals of Epipactis helleborine, the widespread common Helleborine, broad-leaved Helleborine, are marked in red. And you can see that they're distributed widely across this evolutionary tree. And what that is telling us is that the majority of species within Epipactis originated from within the widespread broad-leaved Helleborine. So broad-leaved Helleborine is a widespread ancestral species that's giving rise to large numbers of often more localized descendant species. And that shows even more clearly if you look at evolution not from the side, but from the top. This diagram is based on the same genetic data, but this time you can see the origin of this group of Epipactis right in the middle of what looks like a fly splattered on the windscreen. And then radiating out from that central point are a whole bunch of closely related species heading off in different morphological directions, different genetic directions. But you'll also see in the middle, marked with the red polygon and the red dots, are all the individuals of Epipactis helleborine. You can see that they represent an ancestral plexus from which these other more genetically distinct species have originated. Part of the reason for this is likely that the Epipactis helleborine populations are more allogamous and are mixing up their genes more. whereas at least some of these radiating species shown in green are likely to be more autogamous, so when they accumulate a mutation, it remains in the population. That's one possible interpretation. So this is beginning to look as though the conventional hypothesis about how these species originate is correct. But if we start to use our genetic information, our RADseq data, to look at levels of inbreeding in these populations, you can calculate inbreeding levels quite readily. And we found that the result was not what was expected. In this diagram, I've shown supposedly autogamous species have no green background. I've picked out the allogamous species in green. And you'll see that really only Epipactis helleborine, subspecies Helleborine, the widespread ancestral broad-leaved Helleborine, has a significantly lower inbreeding coefficient than the rest. All of the rest are showing very high levels of autogamy, of self-pollination, self-fertilisation. So this supposed distinction between allogams and autogams isn't really holding up in the face of the genetic data that we've acquired. The autogams aren't quite as autogamous as one might have hoped. And the allogams are certainly nowhere near as allogamous as one would have predicted, with the arguable exception of Epipactis helleborine, subspecies Helleborine, over here on the right. So the next question we might want to ask is, how does Epipactis compare with other groups of British orchids? And that's what I've done here. I've picked out a few random examples of British orchids, Cypripedium, Cephalanthera, Orchis and Anacamptis, all of which have had their inbreeding coefficients calculated. And you'll see that all four of these species have inbreeding coefficients lower than 0.2. In other words, they're almost completely allogamous. Almost all of the pollinations, successful pollinations that take place in these species do so between one plant and another plant, not within the same plant. So it does look as though Epipactis is particularly keen on self-pollination. Even the so-called allogamous, cross-fertilizing Epipactis helleborine has a fairly high level of autogamy. And we really need to find an explanation as to why autogamy is so popular within Helleborines. I think the answer lies in this kind of footage. Here you see a bee working its way systematically up a spike of Epipactis palustris. See how it visited several flowers on the same spike before finally jumping across to an immediately adjacent spike. And these spikes are probably from the same genetically identical individual anyway, because Epipactis palustris is rhizomatous. So the bulk of the visits that this insect is paying are to different flowers on the same inflorescence. And of course, all the flowers on a single inflorescence are genetically identical. So putting pollen from the stamen to the stigma of the same flower is not the only way to achieve autogamy. You can also achieve it by moving pollen between flowers on the same inflorescence, and that's known as geitonogamy. And I think because Epipactis has relatively large inflorescences with substantial numbers of large flowers, I think it's particularly prone to geitonogamy, which reduces the amount of genetic variation present amongst the Helleborines. Another question we might ask is, you would predict that a genuinely autogamous self-fertilizing inflorescence, all the flowers would set seed, because each flower would pass pollen from the stamen to the stigma, from the pollinium to the stigma. Whereas in a cross-fertilized spike, you would predict that at least a substantial proportion of the flowers would not get pollinated because insect pollination is not sufficiently efficient. Well, here I've taken a large number of studies of fruit set in different Epipactis species and summarized them in this right-hand diagram. And again, I've marked the supposedly cross-fertilizing species in green and left the self-fertilizing species not highlighted. And essentially, there's very, very little difference between these different species. They all set fruit, they set seed in about 80% of the fruit. So there's nothing here to suggest that there is a radical difference between autogams or allogams, or that one of these reproductive modes is more successful than the other. And you can actually take this kind of experimentation further by bringing some of these species into cultivation and deliberately either cross-fertilizing or self-fertilizing the flowers. And when this was done by some of my Belgian colleagues for 5 species here, 3 of which are cross-pollinating, 2 of which supposedly self-pollinating, they found that there was no meaningful difference in the proportion of viable seeds in the different fruit. And although fruit set in some of the species was lower when you deliberately selfed it or left it to see whether it's selfed in the wind, the difference, in my opinion, is not great enough to actually suggest that one of these strategies would be more successful than the other. I think this is a very subtle distinction. And I don't think the amount of seed that you produce is of great importance when an individual capsule contains several thousand individual seeds, and there are many successfully fertilized capsules on any one inflorescence. The amount of seed produced by a typical orchid is absolutely phenomenal. You're talking millions for the lifespan of each individual orchid seed. And of course, only one of those seeds needs to establish itself to replace the parent and to keep the population stable. One question we might ask is just simply look at the distributions of these different species within the British Isles and say which are the relatively successful widespread species invading a range of habitats. And it won't be a surprise to any of you to see that the broad-leaved Helleborine, Epipactis helleborine here on the bottom left, is the most widespread throughout Britain other than in Scotland, other than in northern Scotland. The species aligned on the bottom of this diagram are all the supposed allogams, cross-fertilizers. The ones on the top of this diagram are the supposed self-fertilizers. And you'll see that you can actually arrange them as kind of pairs of taxa, neither one of which is more successful than the other. So in southern England, we have purpurata and phylanthes with roughly equal numbers of grid squares occupied. For atrorubans and junensis further north, roughly equal numbers of grid squares occupied, and even for palustris and leptochila. Palustris, of course, relatively widespread, but confined to damp habitats. So there's no real evidence that being an allogam is any more successful than being an autogam. The only thing you can say is that Epipactis helleborine is more successful than the remainder. So if we try to sum up this part of the talk, gytonogamy, along with other non-genetic influences on the gynostemium, is responsible, in my opinion, for an allogamy-ulogamy continuum, and one that is skewed towards autogamy. So if we just list the factors affecting reproductive mode in more detail, first of all, I think the arid environment of some of these species, like junensis in particular, is of considerable significance. In an arid environment, the pollinarium gets desiccated and the pollen disaggregates. It also desiccates the vesidual glue. That little glob of glue dries up more quickly in an arid environment. And once that glue has set, it's much easier for pollen to pass from the pollinium to the stigma of the same flower. And it also increases the risk of the flower not opening properly, at which point its only option is to self-pollinate. Also, development, ontogeny is important. The anther apex continues to grow as the flower opens and remains fertile. And that pushes the pollen mass outwards and downwards towards the stigma of the same flower. So the longer the pollen mass remains uncollected by a visiting insect, the more likely it is to contact the stigma of the same flower and cause autogamy. And then lastly, and the focal point of this part of the talk, is geitonogamy, which is pollination of different flowers on the same inflorescence, which has the same genetic consequences as pollinating the same flower. And that's encouraged by having a many-flowered inflorescence, so the insect has to work systematically up that inflorescence. A many-inflorescence rootstock, and these Epipactis all have rhizomatous growth, so many of them produce multiple inflorescences. You get a crude reorientation of pollinaria while the pollen mass is carried by an insect, whereas in other more advanced orchids you get a much more effective system of reorientation, in orchis, for example. The diversity of pollinators is encouraged by the nectar reward that these Epipactis produce. They produce sufficient nectar in some cases for it to actually cause the insects to become drunk. And there are long flight distances in low density populations, which is something that you often find with Epipactis. So there are lots of different contributors to having a high level of autogamy. And I'm not convinced that this is of great importance in terms of determining whether these species speciate or not. And I actually think that at least as important is their ability to invade different habitats and different soil types. It might be that we're actually looking at the wrong end of an Epipactis plant, and that its relationship with mycorrhizae could be of greater significance. There's been a lot of elegant research recently showing how Epipactis plants actually draw on adjacent trees by using mycorrhizal fungi networks in the soil that are linked to both the adjacent trees and the orchid. And the orchid can use that system to download nutrients from trees into its own rooting system. Now, many generalist orchids, like orchis, for example, that they rely almost entirely on photosynthesis for their nutrition. And therefore they don't concentrate particular isotopes of either carbon or nitrogen. But you can estimate how much an orchid is tapping into the adjacent trees by seeing out how much of these different nitrogen and carbon isotopes they accumulate. And that has been done in this case by Shebold et al. For the genus Epipactis, for several species within Epipactis. And very close to the zero line, the crosshairs, if you like, in this diagram, we have Epipactis palustris, which is telling us that it doesn't rely very much on mycorrhizal fungi. It's mostly photosynthesis that provides nutrition for Epipactis palustris. But as you work your way up through Epipactis helleborine, towards the top of this diagram, the top right of this diagram, through Helleborine and purpurata into various forms of Leptochila and Junensis, you see that more and more of the nutrition in these orchids is coming from adjacent trees. And interestingly, at the same time, the preferred habitat of those orchids is becoming shadier and shadier. There's less and less opportunity for photosynthesis. So I think that it may well be changes in relationship between orchid and mycorrhizae, or genetic changes that allow the orchids to better exploit nutrition from adjacent trees that allow them to invade new, more challenging habitats. And it's probably through that mechanism that it must at least play some part in the origin of these multiple species that have all speciated from within Epipactis helleborine. Okay, that's some pretty high level science. Now let's move on to something a little easier to grasp that looks at these orchids from a Northumbrian perspective, and in particular asks the question, just how many species of Epipactis are there in Northumbria? Because a lot of description of new taxa has been based on Northumbrian material. And here's our first example, Epipactis youngiana, which was always regarded as being closely related to Epipactis helleborine, but it looked as though it had features that made it more autogamous than typical Epipactis helleborine. And this diagram is actually based on the protein work that I showed you at the beginning of this lecture, but the principles still apply. And here we're comparing plants of Epipactis helleborine with plants of Epipactis, inverted commas, youngiana, from the same population of Epipactis. So these are mixed up together, but on the basis of their morphology, we're saying that some of these plants are helleborine and some of these plants are youngiana. Now, if youngiana were genuinely a different species, you'd expect all of the youngianas to cluster together and all of the helleborines to cluster together. But that isn't what you find. What you find is that Helleborine from settling stones clusters with youngiana from settling stones, and Helleborine from Bardijksbeen clusters with youngiana from Bardijksbeen. And what's more, they're genetically very similar, or in the case of the Gosforth Park plants, identical. What this is telling us is that these are single populations of single species. What was being described as youngiana is just a minor morphological variant of the widespread Epipactis helleborine. So that's one taxon that was regarded by quite a few taxonomists as a subspecies of Helleborine, and by others, a small number of people, as a full species. But it certainly doesn't qualify as either a species or a subspecies based on this evidence. So I think we can scrub youngiana. Let's move on to discuss Epipactis tainensis, first described in the Tyne Valley in 1973, first viewed as a form of Leptochila, but later described as a subspecies of Junensis. Well, if we enlarge the Junensis part of this high-level evolutionary tree, phylogenetic tree here, look at that in more detail. This is the Junensis portion of the tree. And two plants of Junensis and one of Tainensis were analysed as part of this project. The two plants of Junensis from the Type region in Lancashire, Tainensis from Alston in Cumbria. And you can see that they're almost genetically identical. There is no question that Tainensis is part of Junensis. And really the amount of genetic distinction is not enough to view it as a subspecies. We could set it up as a morphologically subtly distinct variety. Something else I could point out at this point is there are several other names in this diagram. Next to Junensis, we've got Rhodaensis and Bugachensis from France, Hungary, Germany, and Austria. What that part of the diagram is telling us is that those two are part of Epipactis junensis. Although continental Epipactis specialists have given them separate names, they are actually genetically, from a genetic viewpoint, near identical to British Epipactis junensis. So until recently, many people viewed Epipactis junensis as endemic to the British Isles, or if not, then only just touching on the continental coastline. Now we can see that junensis has been masquerading under other false names on the continent and stretches eastwards at least as far as Hungary. What about Epipactis subspecies, Helleborine subspecies neerlandica? This has long been recognised as occurring on the Dutch and Danish coasts, but as it was described in the 1930s, but as early as 1952 it was reported from South Wales, although this idea didn't become widespread until this century. I was highly sceptical that the South Wales plants had anything to do with the plants on the Dutch and Danish coasts. It seemed obvious to me that they were a minor local variant of Epipactis helleborine that had cropped up on the dunes at Kemfig in South Wales. But if you actually look at the relevant part of our DNA-based tree here, you'll see that Neerlandica, and Rentzii is just a minor variant of Neerlandica here. Here are the Danish Neerlandicas at the bottom of that tree. Just above them is the Dutch Neerlandica sample that we analysed, and then above there is a Kemfig sample of Neerlandica that we analysed, and a plant that we attributed to Epipactis helleborine, also from Kemfig, and they form a close-knit group. They're obviously part of the same subspecies. But what's really interesting is, yes, the Kemfig plants really are very closely genetically similar to the Dutch and Danish plants. And it seems likely that in relatively recent past, seed has been carried in airborne currents from the Dutch or Danish coastlines to Kemfig. And based on this evidence, it's probably from the Dutch coastline rather than from the Danish coastline, because the Dutch plant is more similar to the British plants. What it also slightly embarrassingly says is that the plant that we identified as straightforward Helleborine from Kemfig was similarly a plant of Neerlandica, even though it was growing in woodland rather than in the more characteristic dune systems. So we've eliminated a couple of species or a couple of taxa that were thought to be British but turn out to not have biological reality. Now we're finding here a plant that I thought didn't have biological reality, but does, based on the genetic evidence. But probably what the question that all of you are asking by now is, yes, that's all very well, but what's the real hot topic around here is the Lindisfarne Helleborine, Epipactis inverted commas sancta. And I have to take partial responsibility for this because I was one of the people who, round about the turn of this century, argued that on the evidence then available to us, this one population on the left-hand teardrop portion of Holy Island did seem to be distinct from any other British population of Epipactis and probably did deserve to be viewed as a separate species. Those of you who have visited Lindisfarne and possibly sought out this species will know that there's a relatively small number of plants just confined to the western end of the island of Lindisfarne. And those of you who've actually looked at these plants and then compared them with populations of Epipactis junensis elsewhere, for example, on the Lancashire coast, will have realised that the differences are not very great. But what I want to do a little mischievously is play taxonomic darts with Epipactis sancta and look at how over the last 25, 30 years, different taxonomists have treated this one population on the island of Lindisfarne. Well, the first person to actually refer in print to this population was a gentleman with the distinctive name of Smith in 1958. His observation was picked up by Epipactis expert Donald Young in 1962, who said, yes, this Lindisfarne population is Epipactis junensis. Richards and Swan followed up with a similar conclusion in 1976. But then continental Epipactis specialist Robach in 1983 declared that this population actually lay midway between two continental species, Epipactis pontica and Epipactis muelleri. Then along came Pierre Delforge, the author of those famous series of field guides for continental orchid species, who in 1995 said, oh no, it's Junensis. Sell and Morel in their Flora of the British Isles went back to the idea of the Lindisfarne population being continental in nature and attributed it to muelleri. Newman and Bucher-Pfennig in 1997 decided that the Lindisfarne population belonged in Epipactis peatsii, which our analysis has demonstrated is a non-existent species. The populations the continentals call peatsii is actually Leptochila. Devillet and Devillet-Terscherin a little later also suggested that it was peatsii, as did Delforge in the second edition of his field guide. But the following year he decided, oh no, maybe not as a variety of peatsii, I'll establish it as a separate species, Epipactis sancta. This is 2002. That same year, Squirrel et al. Provided the first genetic analysis that included Lindisfarne plants and said, no, actually, it may well deserve to be a full species, but its closest relative is Junensis, not Leptochila, not peatsii, not muelleri. Kreutz ignored the genetic evidence in 2004, made it a subspecies of Leptochila. Stace in the, I think it's second edition of his Flora of the British Isles, stuck with muelleri. Then Delforge in his third and fourth editions continued to treat sancta as a full species. Hollingsworth et al. And my good self in 2006 stuck with the genetic evidence and said, no, it really does look as though sancta is a full species close related to Junensis. Kreutz then had another crack at sancta and decided to make it a subspecies of Junensis after all. And then Richards and Squirrel stuck with my original argument that sancta is a separate species close to Junensis, but expressed some legitimate concerns about the idea. Before Stace, in the next two editions of his Flora, followed, again reluctantly followed my suggestion and kept sancta as a full species. So probably the question you're asking now is, where would I put it today? And the answer is back where it was in 1958 as possibly a subspecies, possibly a variety of Epipactis junensis. Why have I changed my mind? Well, not because we included material from Lindisfarne in our high-level genetic analysis. There are no Lindisfarne plants in this evolutionary tree. It was because the evidence, the genetic evidence on which we decided that the Lindisfarne plants look like a distinct species was very limited. We're going back 20 years. And I'll show you the evidence on which we made that decision to separate the Lindisfarne plants from other populations of Epipactis junensis. That is it. There's a representative individual of Junensis, Sancta, and Tainensis, just for one gene in the chloroplast of these orchids. And you'll see that Junensis and Tainensis have identical sequences of bases here, A, C, G, and T. The only difference with Sancta is it's got a few letters missing. And those letters that are missing would have been removed as a single event, a single chunk. So there's just one small genetic change separating Sancta from Junensis. And at the time, all we could do was go with the evidence that we had and say that is a distinct species. Whereas now we can sequence pretty much the entire genome. We can sequence the whole of the plastid genome, all 150 plus thousand base pairs, and we can get even more information out of the chromosomal, out of the chromosomes, out of the nuclear DNA. So this kind of analysis is basically yesterday's analysis. And today, seeing that the difference is so small, even though I don't yet have a high-level analysis for the Lindisfarne plants, I'd be willing to stake a significant sum on these being a minor morphological variant of Epipactis junensis. So, where does that leave us? Can we pull a few major themes together out of the last 49 minutes? Well, firstly, genetics. I hope I persuaded you that genetics is an increasingly powerful tool for circumscribing species and interpreting evolutionary mechanisms. One thing I haven't really focused on until now is I haven't presented you with any morphological analyses. All of those taxonomic decisions that I talked about with respect to sancta was based on people either looking at dried specimens or turning up on Lindisfarne, taking a quick look at the population, hopefully looking at a few populations of other species as well, and saying, oh, it looks different. That's not the kind of work that we should be doing. There should be a lot more effort made to analyse the morphology of Epipactis. And indeed, until COVID, that's exactly what I was proposing to do, starting on Lindisfarne. And with luck, I still may be able to do that this coming field season. But it's just striking how much more complete and compelling at the present time the genetic information is than the morphological information. Secondly, in my opinion, traditional taxonomists continue to oversplit Epipactis, particularly on the continent, whereas we've got reasonable stability in the British Isles. And the irony being that after a lot of chopping and changing, we're recognising the same number of species that were recognised in the mid-20th century. Having said that, so there have been circumscriptions changing, not least with sancta, which has been some sort of botanical pinball bouncing between different species. At least we can now place particular populations and particular morphologies with confidence in their correct species. I was really pleased to see that several genuine Epipactis species, some British, some continental, did originate within Epipactis helleborine. Those include Epipactis junensis and possibly Epipactis leptochyle. And it's also interesting to see that the group is actively speciating. It may well be that in the not too distant future, Epipactis helleborine neerlandica is sufficiently distinct to be recognised as a species in its own right. I only hope there are still taxonomists alive to make that decision when it happens. And lastly, arguably all Epipactis species have dominantly autogamous fertilisation. The idea that there's this bimodality between autogams and allogams simply isn't true. And I think a major part of the reason for that is frequency of geitonogamy, of insects working their way systematically up the inflorescence of the same plant, consequently preventing the development of mixing of different genomes. And that's something, of course, that any field botanist can actually sit there and observe, or indeed field entomologist. It would be really nice to get better data on those kind of observations. But I do think that we probably need to seek alternative explanations for the contrasts amongst the Epipactis species in their degree of ecological success and indeed in their ecological preferences, their habitat preferences. So with that, I'll just thank you for listening. I hope that this wasn't too technical. I hope that at least you got some of the major themes out of this and can see the kind of direction that this sort of study is taking. The world is going to remain a pretty exciting place as long as these kind of observations and interpretations are still being produced. I'm sorry that the format of the lecture doesn't allow me to take questions, but I did put my email address on the first slide of this lecture, and I'll be happy to correspond with people who have serious questions or indeed are interested in developing their own modest research project. With that, I thank you very much indeed.